So the learning outcomes for today's video, what I want you to learn about regarding the categories of amino acids and primary structure of proteins, is I want you to be able to identify the category an amino acid belongs to by looking at that amino acid's R group. I want you to be able to properly read and interpret table 3.1 of your textbook, and we'll look at that table a little bit later. I want you to be able to predict how the chemical environment of a proteinatable group might change its pKa. I want you to be able to draw a peptide bond between two amino acids. And finally, I want you to explain the relationship between the primary structure of proteins, the other three levels of protein structure, and protein function. All right, first, when we categorize amino acids, we categorize them solely based on the nature of their R groups. Okay, so we're not looking at the alpha amino, we're not taking into consideration the alpha carboxyl group, we're only looking at the R groups. And the first category are the nonpolar aliphatic R groups. Those are going to be your R groups that are essentially hydrocarbons. Carbons and hydrogens, carbons and hydrogens, carbons and hydrogens. So those are going to be the ones then that are nonpolar, because remember, carbons and hydrogens have about equal electronegativity. There's a sulfur in here, but here's methionine that's got a sulfur, and even though sulfur is technically a fairly electronegative element, it is methionine is still considered a non-polar amino acid. Then we have the aromatic amino acids. Those are the amino acids who in the R group have aromatic rings: phenylalanine, tyrosine, tryptophan. And these are special because they have this property where they absorb a very specific wavelength of UV light. Notice when you shine various wavelengths of light and then measure the absorbance, they kind of peak right around 270, 280 nanometers, the absorbance peaks. And scientists use this as a way to measure the concentration of a protein in solution. They can take that protein, they can put it in a spectrophotometer, set it at about 280 nanometers, and figure out the absorbance, and then they can run a few calculations, and from that they can figure out the concentration of that protein. So this is a pretty useful property of these aromatic amino acids. Then now we go into our polar but uncharged R groups. And we've got, those are going to be the ones that have these functional groups: hydroxyl group, sulfhydryl group, asparagine and glutamine have these two groups here. And I should say that when you look at table 3.1, you will see that cysteine is, the sulfhydryl group is deprotonatable. It can donate that proton. Same thing with this serine here. The difference is that under physiological pH, it really doesn't happen. Okay, you've got to change the pH beyond physiological pHs to get this hydroxyl group to deprotonate, or this sulfhydryl group to deprotonate. Cysteines, so if we take a look at the cysteine with the sulfhydryl group, have this special property that's going to become very important once we start talking about protein structure. And that is, when you get two sulfhydryl groups adjacent to each other, there is a redox reaction that occurs where we lose our protons, we lose some electrons there, and that forms a disulfide bridge, a covalent bond between these two sulfur atoms. And again, this is a interaction that stabilizes the structure of many proteins. Now we get into our positively charged R groups. These are amino acids that under physiological pH, so around 7.2 or so, this is the way they exist. They exist in this protonated state, and histidine is right on that edge. Histidine, the pKa of this R group here is 6. And so it really depends upon what the actual pH is of a solution, but it can be protonated and carry a positive charge. Lysine got this amino group here. It's protonated. It's got the positive charge. And then, of course, you know what amino groups can do. They can be deprotonated or protonated depending upon the pH of the solution. There are then negatively charged R groups. Aspartate, glutamate, also called aspartic acid, glutamic acid. When the, here it is. When the R group, so the reason aspartate is a negatively charged R group is because it has a carboxyl group. Notice this is a second carboxyl group here. We have our alpha carboxyl group and the carboxyl group in the R group. We have an alpha carboxyl group here in glutamate and one in the R group. Aspartate is what we call it when it is deprotonated. Aspartic acid is what we call it when it's protonated. Okay, so here's that table. I believe table 3.1 from your textbook. It's something that you're going to have to want to really study. You won't have to memorize anything in this table. You'll just have to know how to use it. But one thing I want to show you here is, let's take a look at the alpha amino group of a bunch of amino acids. And we're looking at the pKa of that alpha amino group. So this is the same alpha amino group, same alpha amino group, same alpha amino group. But notice, depending upon which amino acid we're looking at, it has different pKa's. So we normally think of a pKa as being something inherent for a particular group, right? An amino group has a pKa of 9.0 regardless, but obviously that's not the case. What this means is the immediate chemical environment of a protonatable group can affect its pKa, and this is a really important idea to keep in mind. When you start reading that journal article, we'll talk more about that, and that'll come into play more a little bit later. So let's give some examples. Let's take a look at this. Acetic acid is like the most fundamental form, a molecule that has a carboxyl group on it, right? It's a carboxyl group stuck to a methyl group, done. And in this environment, this carboxyl group has a pKa of 4.8, okay? Now let's change the chemical environment. Let's pluck off one of these three hydrogens and add to it an amino group. So we've changed the immediate chemical environment of that carboxyl group. We've added very close to it a group with a positive charge. So let's think about that positive charge. Two things are going to happen here. This hydrogen is partially positively charged. There's going to be repulsion between this positive charge and this partially positively charged proton here. So it's going to make that carboxyl group, it's going to make it easier to kick off that proton. It's going to essentially make the carboxyl group have a lower affinity to that proton. In addition, once that proton is kicked off, that leaves the carboxyl group with a negative charge, and now this positive charge and this negative charge stabilize each other. So when you stabilize something, you make it more likely. You give it a lower energy and you're making it more likely. So this kicking off the proton is something that's happening. And then on top of it, once the proton's gone, we have this positive and negative charge that stabilize each other. So that in essence, what's happening is we're making it much easier for the carboxyl group to be deprotonated when we compare it in a situation where it's not very close to an amino group. So that means between the repulsion going on here and the stabilization going on here, that we are making it easier for that carboxyl group to lose its proton, which means we are lowering its pKa. And sure enough, when you measure the pKa of this carboxyl group in this chemical environment, it's 2.34. That's a pretty big difference. And again, it's because of the chemical environment. All right, let's take a look at another example. So here we have an alanine amino acid. And we have, here is the alpha amino group, here is the alpha carboxyl group. And again, just like we sort of paid attention to, or that we discussed in that previous slide, is that the carboxyl group's deprotonated state is stabilized when it's next to a protonated amino group. So what that means is, in this particular situation, we are stabilizing the deprotonated state of the carboxyl group and the protonated state of the amino group. So we should have the lowest pKa for a carboxyl group and the highest pKa for the amino group. Now, let's do this. Let's connect a bunch of alanines together, and then let's look at that alpha amino group here and that alpha carboxyl group here. Now notice, look how far they're separated. So that means there is more space separating this group from this group. There isn't going to be as strong an interaction between this alpha amino group and this alpha carboxyl group. So that interaction that's occurring here is going to become weaker and weaker and weaker and weaker. So let's think about that. If that interaction is becoming weaker, then there is nothing stabilizing the deprotonated state of this carboxyl group. It should now be able to hold on to protons a little bit better, so its pKa should go up. Let's take a look. 1 alanine, 2.34. 2 alanines separating the two groups, 3.12. 3 alanines, 3.39. 4 alanines, 3.42. Yes, this data demonstrates that when you separate these two groups, that interaction becomes less and less and less and less, and therefore the pKa of this carboxyl group goes up and up and up and up. Let's look at the amino group. So the alpha amino group, its protonated state is stabilized by being next to this carboxyl group. In essence, what we're doing here is, because of this chemical environment, we are increasing the pKa of the amino group, making it better at holding on to its protons, which means when we separate these two by connecting a bunch of alanines together, the pKa, this interaction will be less and less and less and less, and the pKa of this alpha amino group should go down and down and down. And let's take a look. So here, it's just the two groups on a single alanine, 9.69. When they're separated by two alanines, 8.3, 8.03, 7.94. Indeed, our prediction is correct, that when you separate these two, that alpha amino group is no longer being stabilized by this negative charge, and so it can't quite hold on to its proton as strongly as it did when the two were very close to each other. Again, this idea that chemical environment can affect the pKa of some sort of functional group will become important when we talk about your first journal article. Okay, there are 4 levels of protein structure, and they all exist simultaneously. The one that we're going to talk about today in this video is the primary structure. Now, that is nothing more than the sequence of amino acids connected to one another via peptide bonds. And it's the primary sequence, or excuse me, the primary structure that has all of the information necessary for that protein to fold into its secondary structure. And again, that primary structure has all the information necessary for that protein to fold further into its tertiary structure. And finally, if the protein is made up of more than one polypeptide chain, each one of those come together in a specific way in the quaternary structure. So all of this is dependent upon the primary structure of the protein, the sequence of amino acids connected by peptide bonds. So, what is a peptide bond? How does a peptide bond form? Well, here is our growing polypeptide chain, our growing chain of amino acids. This is the alpha carboxyl group. And then here's our incoming new amino acid. This is the alpha amino group. We lose the OH from the carboxyl. We lose an H from the alpha amino. That's our H2O. Right? That's a dehydration reaction. And then what we end up with is a covalent bond between the alpha amino group nitrogen, the alpha carboxyl group carbon. Right there is the peptide bond. We still have a carbonyl oxygen there, and we still have a hydrogen here. This is the peptide bond. And now this is the new alpha carboxyl group here that we'll add on to, we'll add on to, we'll add on to. And so what you end up getting is you get a string of amino acids that you can keep adding to, adding to, adding to. And from that string, from that backbone of peptide bonds, you get the R groups, the R groups, the R groups, the R groups. There's a term that you will read in maybe the book, maybe the journal article called residue. And a residue is what we refer to as an amino acid when it is in a polypeptide. And really what that's indicating is that an amino acid outside of a polypeptide has an extra water associated with it. Whereas when that amino acid is in a polypeptide, it's missing that molecule of water because of that dehydration reaction. Polypeptides and proteins, those two terms are kind of synonymous. You may hear me say either one. Protein is when the molecular weight is over 10,000. Polypeptide, we usually talk about it when it's below 10,000 of molecular weight. If we have two amino acids connected together, we would call it a dipeptide. If three, tripeptide. Oligopeptide is several amino acids connected to each other. Every polypeptide, no matter how long it is, will always have one free alpha amino group. Therefore, we call that the amino terminal N or the N terminus. And then we will have one free alpha carboxyl group. We call that then the carboxyl terminal end or the C terminus. Proteins come in all sizes. You don't have to memorize any of this. If we just take a look at the number of residues or the number of amino acids, you can have some relatively small ones, 104 amino acids, and then the human titan protein, 26,926 amino acids, huge. You can have differing number of peptide chains, right? Glutamine synthetase has 12 different polypeptide chains that come together to make up the whole protein. If you want to get a general idea of how many amino acids or how many residues are in a protein, take the molecular weight of that protein, divide it by 110. 110 is about the average molecular weight of an amino acid. Proteins have different amino acid makeup. So there's nothing to say that every protein must be 10% glycine. So if we take a look at the percentage of serine in two different proteins, here 11.4%, here just 1%. So different proteins have different amino acid makeups and different amino acid sequences. Proteins will also have stuff connected to them. So after they've been translated, there will be things that are covalently bound to them. There are lipoproteins that have lipids bound to them. Glycoproteins have carbohydrates. Phosphoproteins have phosphate groups. Heme proteins have heme groups. Flavoproteins have flavin nucleotides. And that is a whole bunch of metalloproteins that require various metal ions. And we're going to see some of these. We'll see succinate dehydrogenase. We'll see hemoglobin. We will see alcohol dehydrogenase. And so you'll see examples of various conjugated proteins, proteins with non-amino acid stuff connected to them. Now, We are going to get to this idea over and over and over again. But let's get a first introduction to you. We've already mentioned that the primary structure, the sequence of amino acids within a protein, is all the information that that protein requires to properly fold into its final structural form. That's it. Just the sequence of amino acids will dictate what that final structure of the protein is. On top of that, the structure of the protein is what dictates or determines the function of the protein. So what that means then, indirectly, it's the primary sequence of the protein that dictates its and determines its function. If you do something to mess up the primary structure, if you change the sequence of amino acids, you will change the structure of that protein and therefore change the function. Let's take a look at a classic example of that. Here we have the hemoglobin protein. And here we have a part of the primary structure, the sequence of amino acids. That's enough information to properly fold it into the secondary and tertiary structure. That's enough to have it stick with the other components to form hemoglobin, and that's enough for these hemoglobin proteins to do what they're supposed to do within a red blood cell, which is to transport and carry oxygen. But if instead the protein has a mistake in it, instead of amino acid 6, it has a valine instead of a glutamate, well then we're going to have a problem. It's not going to fold properly. It's not going to interact with the other polypeptides properly, and therefore, because it doesn't have a proper structure, it's not going to have its proper function. It'll have a new function. It sticks to one another. Stick, stick, sticks. They're not supposed to do that. But because of this change, because of this structural change, we get now a functional change. They stick together. They form these aggregates, these fibrils. They distend, and misshape the red blood cells, and then that causes a brand new trait, and that trait being sickle cell disease. So, structure of a protein, the sequence of a protein gives rise to its structure. The structure gives rise to its function. If you change the sequence, you change the structure, and therefore you change the function. That idea will become increasingly important as you start reading Making of the Fittest, as you start reading the journal article. All right, so again, review this lecture and make sure that you can identify an amino acid the category of an amino acid based on that amino acid's R group. Make sure you can properly read and interpret Table 3.1. Be able to predict how a chemical environment of a proteinatable group might change its pKa. Be able to draw a peptide between two amino acids. And finally, explain the relationship between the primary structure of proteins, the other 3 levels of protein structure, and protein function.